form. Let's move on. I beat this to death already, but polypeptides fold maintained due to amino acid stickiness, and that's from the side chain intermolecular forces. You'll be responsible for identifying what type of intermolecular forces are occurring between the side chains. Are they covalent bond, hydrogen bond, dipole-dipole, or ionic forces? Now, instead of saying the word ionic forces, we use the term electrostatic interactions. Another word for the same thing. If you put ionic forces, I'd still give you credit for this. So, we're assuming that this polypeptide is in its aqueous form and it's folded up properly. Here is a model in this hard to see picture right here. This is a model of a cold shock protein, cold shock polypeptide. This is a polypeptide. Think of this as a molecular EMT, and its purpose is to go around and fix proteins that have been too cold, so to speak, and consequently they start to fall apart. So they run around and start fixing other proteins to make sure they're working properly when your cells get too cold, such as when you're starting to freeze. So we call these cold shock polypeptides. Ones that are bigger than 50 amino acids are cold shock proteins. Pretty cool, huh? So those are the medevac EMT-like proteins which help other proteins which are traumatized due to coldness. You're responsible for memorizing these four: dipole-dipole, hydrogen bond, London force, and hydrophobic interactions, covalent bonds, and electrostatic interactions. I want to focus on these two right here, hydrophobic and electrostatic. So let's go ahead and focus on this one right here, hydrophobic, because that's probably a brand new concept, and the other ones we'll just review. Hydrophobic. So I could have a polypeptide that looks like this, and I'm just going to draw it wrapped around in a circle. The side chains, which are going to be in blue, would look like this right here. And right now we've only studied leucine. So this is leucine. Obviously this is a really crude model. So, for hydrophobic interactions, basically the nonpolar side chains are attracted to other nonpolar side chains. Via London forces. So in this case, via London forces, aka dispersion forces, the nonpolar side chains, they're attracted to each other, like attracts to like. On the same token, nonpolar side chains are repulsed away from polar water. So notice if we have water on the outside, the nonpolar side chains are going to be attracted to each other, and they're going to be repulsed, and I'll go ahead and I'll use green. They're going to be repulsed, and that's my repulse. They're going to be repulsed away from the water. So they're attracted to each other, like attracts like, nonpolar attracts nonpolar, and they're repulsed away from the polar water molecules. And they're repulsed away from the nonpolar, I'm sorry, let me say this all over again. I know I'm going really fast. The nonpolar side chains are attracted to other nonpolar side chains. So the nonpolar side chains at the same time are repulsed away from the polar water molecules. Because they're repulsed away from the other polar molecules, we call these hydrophobic interactions. Why? Because they don't like water. They like their own kind, but they don't like being near water. So that's hydrophobic interactions. There's also another effect, which we'll get into in a bit, called the hydrophobic effect. And this is where water doesn't want to be around those nonpolar side chains, so much so that it actually pushes the nonpolar portion away from itself. And in truth, what actually happens when you start folding these proteins is that you have what's called the hydrophobic effect, where water molecules, because they want to be away from those nonpolar portions, they'll actually push away the nonpolar portion and they'll tighten it into a ball, so that way it minimizes the polar-nonpolar interaction. Pretty cool, huh? Okay. So let's go ahead and keep going. Electrostatic interactions. Let me go ahead and pull out my pen. Electrostatic interactions. Here is another brand new one. Well, not that new. So I'm going to go ahead and create a backbone of our polypeptide, and then I'm going to go ahead and highlight key amino acids. So in this case right here I have a, I don't like how that looks. Let me do it again. So in this case we have a aspartic acid, and then over here we have a lysine. So aspartic acid here we have a lysine. First thing I want to point out is we're always going to assume when we draw these as polypeptides that they're going to be aqueous. So the first thing you want to keep in mind is when you're drawing those polypeptides, you need to go ahead and remove the hydrogen on the carboxylic acid. This goes ahead and makes it a negative charge. You then want to go ahead and put on another hydrogen on the, I'll go ahead and I'll use a black pen for that. You'll go ahead and put on another hydrogen, so it's now NH3 with a positive charge. Now, ionic forces is very straightforward, aka electrostatic interactions. its opposite charges attract. So the positive charge from the lysine is going to be attracted to the negative charge of the aspartic acid. So usually we draw a dash showing this particular attraction. Now, it turns out that if these are really close to each other, within touching distance, then we call these salt bridges. Salt refers to the fact that you have opposite charges which are forming a crude ionic bond. Just as you had positive sodium and negative chloride forming an ionic bond, you have positive lysine and negative carboxylic acid forming an ionic bond. So we call these salt bridges. If it turns out that those amino acids are very far away where they can't touch, it turns out that you can have long distance opposite charge attraction. So you have long distance electrostatic interactions. These long-distance electrostatic interactions, you can think of them as these really long tethers where they're attracted to each other, so the tethers or rope help cohesively keep the protein together from long distances. All right, so we summarize the major types of forces that you haven't covered in the past. Now let's go ahead and review ones you have. Whenever we have covalent bonds, in this case we only study one type of covalent bond that's very, very common, and we call these disulfide bridges. These are also known as cysteine bridges. So we never really use the term covalent bonds, we use disulfide or cysteine bridges. Why? Because they're two sulfurs that come together from two different cysteines to form a permanent cysteine bridge. So we talked about cysteine bridges when we talked about hair and how various components, various strands within the hair itself actually bond together via cysteine bridges. We then talked about salt bridges. We just talked about that now. Here we talked about the hydrophobic effect, also known as London forces. In this case, we have two phenylalanines represented as PHE, and they're attracted to each other. And so these two phenylalanines are actually on top of each other. So these would be hydrophobic interaction or London forces. And finally, if we have two serines, these are hydrogen bonding. Hydrogen bonding. This one I could give you, this one I could give you, this one I could give you, this one I would not give you. Why? Because we have not talked about phenylalanine and what it looks like. So at this point, I want to go ahead and stop right here and go back a couple slides. So I know I'm jumping around quite a bit, but please stay focused. You are responsible for memorizing You're responsible for memorizing these 4 amino acids. There are 20, technically there are 22, 2 of them are kind of weird, but we're not going to get into those. There are 20 amino acids. You're only responsible for knowing 4 of them. You need to memorize these 4. You're then going to apply these 4 amino acids into actual polypeptides. Polypeptide formation with side chain intermolecular forces. So using those 4 right here, at some point I'm going to ask you to sketch a much better accurate model of something that looks like this, showing the various side chains as well as their intermolecular forces. Here we have, let's keep going though for the time being, and I'll go into details of what I expect for those polypeptides. Here we have what's called hydrophobic packing. Again, I talked about how the nonpolar amino acids, they like to be attracted to each other via London or dispersion forces, and the outside water molecules, they only like to be attracted to polar side chains. So for example, they'll be attracted to polar amino acids. However, it turns out that as far as the nonpolar goes, they'll actually push the polypeptide together, and they'll push it together such that the nonpolar portion is right in the center. So that way the water molecules never have to deal with direct, in theory, direct nonpolar-polar interaction. So if you take a look at the example on the right-hand side, here we have the before, and the water molecules, they're attracted to the polar side chains, and then they're pushing together their polypeptide so that the nonpolar portion is right in the center of the polypeptide or protein. Pretty cool, huh? Because they're pushed together like this, this is called hydrophobic packing. You'll have to give me a general definition or diagram in your own words. Here is revisiting the disulfide bridges. So we start off with 2 aqueous cysteines. So in this case, here we have our cysteines, and then they're going to go ahead and form a cysteine bridge, aka disulfide bridge. What happens is the hydrogens come off, and then the remaining electrons that were connected to those hydrogens, they form a brand new covalent bond, aka disulfide bond or cysteine bridge. So more than one way to say the same thing. Disulfide bond, disulfide bridge, cysteine bridge, all of them are correct answers. The enzyme responsible for this oxidation gives off an oxygen, so it actually forms a brand new water molecule. So this is actually a type of condensation reaction. So in this case, here we have actual polypeptides. This is vasopressin. It's an antidiuretic, so it keeps you from urinating. And then this is oxytocin. It does the opposite. It causes uterine contractions. I'm not going to go into detail on those. I'd rather just focus on the pretty molecules. And above all, we know that there is a cysteine bridge present. Pretty cool, huh? Here, I beat this to death already, but you can already tell this is a dry amino acid. This is a aqueous. It's been dissolved in water, such as within a cell or in the blood or body fluid. So in this case, here we have the loss of a hydrogen, so it's a negative charge. Here we have a positive charge for the lysine. I beat that to death already. Here is what a picture of a aspartic acid and lysine looks like. This is my attempt to make it a three-dimensional structure. If you look at it long enough, you may go cross-eyed, so don't worry about it. This is what it looks like from a ball and stick type model, aspartic acid and lysine. I beat this to death already, but here is a poly. Actually, it's greater than 50 amino acids. That's a protein right here. That's actually a heat shock protein. So just like the cold shock protein, the heat shock proteins, they're responsible for providing chemical first aid to near-death proteins which are falling apart because it got too hot. Pretty cool, huh? Proteins helping other proteins. I studied the cold shock and heat shock proteins a lot in graduate school, so that's why I love these particular models. In this case over here, if we were to sit there and I know it's hard to tell, so let me go ahead and use white. So I've circled this in white right here. If we were to sit there and blow this up, you could see a salt bridge because you have a negative amino acid and a positive amino acid. So there are many other amino acids. You're just covering 4. There's actually 20. Well, technically 22. We'll get into that. But so this is actually not aspartic acid. That's actually glutamic acid. But my point in this is an example of a model of a salt bridge on a protein. And the purpose for this is to keep the protein together. All of these intermolecular forces on these side chains make the polypeptide very sticky, keeping it close together. Here are the long-range electrostatic interactions, which I talked about. Here are the long-range electrostatic interactions. Pretty cool, huh? I love those. Love. So you can take a look at those. The positives, for example, would be, let me use a white pen. I'll use what stands out? Nothing stands out. I'll use a white pen on these. So the positive, think of those as lysines. The negative, think of those as aspartic acids, which lost their hydrogens. So we have our positive lysines, you have our negative aspartic acids. Consequently, from long distances, you will have opposite charge attraction. Now, it turns out that the positives and negatives from multiple positively charged amino acids, positive and negatively charged amino acids, will have long distance interactions. So think of these as these long invisible tethers which help keep the protein together. Now, here is a crude model summary showing the different positive and negative charge interactions. And basically, because it occurs over multiple amino acids, think of this as the overall electrostatic average from each of these multiple positive, negative charged interactions. So basically, the red means excess negative, the blue means excess positive, white means neutral. So what we found, not always, but we found in nature is nature prefers to have the positive and negatives spread out equally. So think of it as a checkerboard pattern of positive and negative amino acids. These positive and negative amino acids tend to be on the outside of a protein. So I'll just go ahead and, label this negative, positive, negative, positive. They tend to be on the outside of a protein. So we're looking on the outside of a protein because like attracts like, the positives and negatives are going to be near polar water. And it tends to be in a checkerboard, so that way all the positive and negatives um, help cancel each other out. Pretty cool, huh? Whenever you have proteins which are considered very unstable, they tend to have an excess positive or negative charge in one area. Consequently, it disrupts the overall protein and makes it easier for it to fall apart. The more stable proteins, which are more resistant to falling apart, tend to have evenly balanced out checkerboard positive and negative amino acids spread out uniformly through the protein. Pretty cool stuff. So this concludes the summary of this portion of the Chapter 9, Part 1 slides.